In 1905, two French Egyptologists were excavating a site on the western bank of the Nile near Luxor, called Gabanat el Khurud, the Valley of the Monkeys. They were not expecting to find anything of particular significance. The site was known primarily for painted tomb walls nearby, and the dig was routine by the standards of early 20th century archaeology. Then they broke into a sealed chamber and found 17 baboon skulls, along with a substantial quantity of skeletal material, some wrapped in linen, some stored in ceramic jars, some placed in wooden coffins. The mummies were cataloged, distributed between museums across Europe, and largely forgotten. Egyptologists noted them. Nobody thought they carried a secret that would take over a century of scientific development to read. The secret was this: those baboon mummies knew where the most mysterious kingdom in the ancient world was located. They had been carrying the information inside their bones the entire time. Baboons are not native to Egypt. They have never been native to Egypt. There is no fossil evidence of a wild baboon population anywhere in the Nile Valley or the surrounding desert at any point in recorded or prehistoric time. Ecological modeling confirms it. The range of the Hamadryas baboon, the species ancient Egyptians considered sacred, runs through the Horn of Africa: modern Eritrea, Ethiopia, Djibouti, Somalia, and the southwestern edge of the Arabian Peninsula in what is now Yemen. The nearest natural population to Egypt is separated by hundreds of kilometers of the eastern desert and the full breadth of the Red Sea. And every baboon that ever appeared in an Egyptian temple, every baboon mummified and deposited in a catacomb, every baboon depicted in 3,000 years of Egyptian art had been brought there from another country, across some of the most difficult terrain and water in the ancient world. And yet the Egyptians had been doing exactly that since the very beginning. A small statue of a Hamadryas baboon inscribed with the name of King Narmer dates to between 3150 and 3100 BCE. Narmer is considered the first pharaoh of unified Egypt. The statue is one of the oldest royal artifacts in Egyptian history. Baboons were there from the first day of dynastic civilization, which means the Egyptians had already established a supply chain capable of transporting living primates across the Red Sea before the pharaonic tradition itself was fully formed. By the time Tutankhamun reigned, between 1332 and 1323 BCE, the baboon was so embedded in Egyptian religious symbolism that the young pharaoh wore a necklace decorated with baboons adoring the sun, and the western wall of his tomb showed 12 baboons representing the 12 hours of the night. The animal was everywhere, in paintings, reliefs, amulets, statues. At Hermopolis Magna, the cult center of the god Thoth in Middle Egypt, the pharaoh Amenhotep III erected colossal baboon statues carved from quartzite, their bodies standing over 4.5 meters high, built to anchor the theological center of a civilization. The question nobody could answer was where all those animals came from. The Egyptians wrote about a place called Punt, a wealthy trading partner reachable by sailing south on the Red Sea, that supplied Egypt with incense, myrrh, gold, ebony, ivory, leopard skins, and living baboons. Records of expeditions to Punt appear from as early as 2450 BCE and continue for over a millennium. The most famous visual record is carved into the walls of Queen Hatshepsut's mortuary temple at Deir el-Bahri, dated to approximately 1470 BCE. The relief shows Egyptian ships arriving at Punt, their decks loaded with goods for the return voyage. Among the cargo visible in the stone are baboons. One sits in the rigging of a ship. The image is specific, detailed, and 1400 years before the Common Era. But despite these records, nobody knew where Punt actually was. No map, no coordinates, no geographic description precise enough to place on a modern atlas. Scholars had debated the location for over 150 years without resolution. The baboon mummies from the Valley of the Monkeys sat in European museum storage while that debate continued. Then in 2020, primatologist Nathaniel Dominy of Dartmouth College published a study in the journal eLife that changed the terms of the question entirely. His team's method was isotopic analysis. Strontium, a chemical element present in soil and water, varies in composition from one geographic location to another. When an animal drinks water and eats food from a specific region, the strontium signature of that region embeds permanently in its teeth during early development. Bone and hair reflect more recent environments. Teeth lock in the birthplace. The team built a reference database of strontium and oxygen isotope compositions from 155 modern baboons sampled across 77 locations spanning eastern Africa and the Arabian Peninsula. Then they measured the isotopic profiles of 7 mummified specimens held in the British Museum and the Petrie Museum, 2 from the New Kingdom period dating between 1550 and 1069 BCE, 5 from the Ptolemaic period after 332 BCE. The results divided cleanly along chronological lines. The 5 Ptolemaic baboons reflected strontium levels consistent with Egyptian origin. They were born in Egypt. Their bones carried the isotopic chemistry of the Nile Valley. This confirmed historical texts from the Ptolemaic era describing captive breeding programs for baboons in the city of Memphis. By the later centuries of Egyptian civilization, the priests had established a domestic supply chain and were no longer making the voyage across the Red Sea for every animal. But the 2 New Kingdom baboons, the older ones, the ones contemporary with the great pharaohs, told a completely different story. Their isotopic signatures did not match Egypt. They did not match any nearby region. The data placed their births in a broad geographic area encompassing modern Eritrea, Ethiopia, Djibouti, and portions of Somalia and Yemen. They had been born in the southern Red Sea region, transported thousands of kilometers by ship, and mummified in a country that had never been their natural home. One specimen, designated EA6736 and held by the British Museum, and appeared to have died so soon after arriving in Egypt that its hair and bone had not had sufficient time to convert to the local isotopic signature of Egyptian water. The animal had been captured somewhere on the Horn of Africa, loaded onto a ship, traversed the length of the Red Sea, and was dead within days or months of arrival. It never acclimated. The isotopic result pointed to a region but not a specific location. For that, a different kind of evidence was needed. 3 years later, Gisela Kopp, a biologist at the University of Constance, attempted something that paleogeneticists had told her was impossible. She tried to extract DNA from mummified baboon specimens. The mummification process, natron salts, plant resins, applied oils, followed by millennia of burial in warm Egyptian sediment, destroys genetic material. Previous researchers had not attempted it because the expectation of failure was too strong. Kopp tried anyway. Her target was one of the baboons from Gabonat El Khouroud, the 1905 discovery, the specimen now held in the Musée des Confluences in Lyon, France, dated to approximately 800 to 500 BCE. She sampled 10 mummified specimens in total. 9 produced nothing usable. The DNA had been chemically erased, but the 10th yielded a complete mitochondrial genome. Mitochondrial DNA is found in thousands of copies per cell, rather than the single copy of nuclear DNA housed in the nucleus. Its abundance makes it the last genetic material to survive extreme degradation. When everything else is gone, fragments of mitochondrial sequence sometimes remain. Kopp's team recovered enough to reconstruct the full mitochondrial genome, but then compared it against a reference database of modern baboon populations from across Africa and the Arabian Peninsula. The mitochondrial DNA does not match populations throughout Africa equally. Different regions carry distinct genetic signatures that have remained geographically stable over thousands of years. The comparison is therefore a geographic fingerprint. The ancient mummy's genome pointed to a population from a narrowly defined area along the coast of Eritrea, not the Ethiopian highlands, not the Somali interior, the coastal strip, specifically the region where the ancient port city of Adulis once stood. Adulis was a major maritime trading hub documented in Greek and Roman sources as a center for the export of luxury goods and live animals, including baboons, to Egypt and the Mediterranean world. The chronological gap between Punt and Adulis had always made scholars hesitant to connect them. Punt disappears from Egyptian records in the early 1st millennium BCE. Adulis does not appear in historical sources until the beginning of the Common Era, centuries later. But the DNA closed that gap with physical evidence. The mummy from Gabonat El Khouroud, dated to 800 to 500 BCE, precisely the transition period between the last recorded Egyptian expeditions to Punt and the later emergence of Adulis as a documented trading center. The animal's genome linked it to the Eritrean coast. The records linked Punt to the same direction. Kopp stated the hypothesis directly. Punt and Adulis may be 2 different names for the same place, used at different points in time by different civilizations. The finding, she noted, only became fully coherent when biological data and historical research were placed alongside each other. This was, separately, the first successful extraction of ancient DNA from a mummified, non-human primate. Every previous attempt had failed. Kopp's method, optimizing extraction protocols for severely degraded material and targeting mitochondrial sequences rather than nuclear ones, produced a result the field had considered unachievable. The techniques she developed now open the question of what other animal mummies in museum collections around the world might still yield genetic information. Crocodile mummies, ibis mummies, cat mummies. Each one a potential data point in the reconstruction of ancient trade routes, animal population histories, and the ecological footprint of civilizations that no longer exist. And the same approach that found Punt in a baboon femur might locate other lost trade partners, other unknown kingdoms, other supply chains that sustain civilizations across distances, and logistical challenges that should, by reasonable calculation, have been impossible. There's one more detail that the science revealed, and it is the one that changes how the entire story feels. The baboon the Egyptians chose to elevate above all other animals, the species they built colossal statues of, the one they associated with the god of wisdom and the scribe of the universe, is not an animal that populations living alongside it in Africa have historically revered. Baboons raid crops. They break into homes. They are calculated and aggressive in their destruction of subsistence farming operations, reducing yields by half for affected families. Throughout sub-Saharan Africa, baboons are largely absent from artistic traditions precisely because they are viewed as pests and threats. The Egyptians, who never shared their landscape with wild baboons, encountered the animal only as an expensive import from a distant and prestigious kingdom across the sea. They saw it fresh, without the accumulated resentment of the generations of crop loss. They saw an animal that sat at dawn with its arms raised toward the sun. They called it divine. The DNA did not change that, but it did specify the coast where those animals were born, the ships they rode to reach Egypt, and the port that connected one civilization's religious imagination to another continent's wildlife. What the science confirmed is that the most sacred animal in the most documented ancient civilization on Earth was a foreigner. It always had been. The Egyptians crossed the sea to find it, paid for it, brought it home, pulled its teeth so it could be handled, housed it in temple enclosures, and when it died, wrapped it in the finest linen available and buried it with the care they gave their gods. Isotopes and mitochondrial genomes read that history from inside the bone. The animal carried its own origins with it through 3,000 years of burial. Scientists compared that evidence to every baboon population on Earth. The answer was a narrow stretch of Eritrean coastline and a kingdom no one had been able to find, now precisely located by an animal that could not speak, but whose chemistry told the entire story.